Okay, good morning everyone, or good day everyone. Today we will discuss with another technique in biotechnology, but we will focus now on the PCR and gel electrophoresis because this is in continuation with the DNA extraction that we've discussed previously. So previously we discussed DNA extraction and we've learned that many people extract this DNA for better understanding and for many applications of the DNA. So this DNA extraction is very useful because it obtains DNA in a relatively purified form which we can be used for further investigations. Let's say for example, we want to clone a gene or we want to do recombinant DNA technology or just even to study DNA itself, it must be extracted first from the organism. So there are a lot of things here we can see on the screen. The importance of or the significance of DNA extraction. It is used for polymerase chain reaction. It will be used for sequencing, for genetic manipulation, for electrophoresis, fingerprinting, and cloning. So this is, or for the DNA extraction, is the initial step towards this uh, activities that will happen later on. And these are the list of activities. So also for plants, this DNA can be, or DNA extraction can be useful, especially if you want to identify something, or if you want to identify, or if you want the gene to replicate in a successive generations of plants. Also for animals, it is also useful. So it helps in the cloning if you want to do cloning or to transfer one animal's DNA to another. And it is a prerequisite for genetic manipulation for PCR to amplify small amounts of DNA sequence of interest so that it can be analyzed separately. Or if you also want to do DNA sequencing, so it is a technique that is used to determine the nucleotide sequence of the DNA. So that is DNA sequencing. For us to know the arrangement of our ATCG or the adenine, thymine, cytosine, guanine, first we need to extract the DNA. We need to have a purified form of our DNA so that we will be able to understand and we will be able to determine the nucleotide sequence. So also DNA extraction is used for forensic studies, paternity tests, ancestry tracking, genetic engineering. There's a lot of things that we can use from the extracted DNA that we created. So once our DNA has already been extracted, so we can use them for further analysis, molecular analysis. We can use, as I mentioned, the PCR, electrophoresis, fingerprinting, and cloning. So that's for the DNA extraction. So now let us move on to the next. So to test the presence and quality of the DNA, of course, after extraction, we need to really know if atong DNA first, if it is in good quality, because that also dictates the success of our experiments. Kung nindot ang atong DNA. So if the quality of the DNA is good, then we can say that there's high possibility of success rate in our further mga lab works. So to confirm the presence and quality of the DNA, we have here two images. So the first image is the spectrophotometer. The second one, kanina ay black and white, is the gel electrophoresis. So this spectrophotometer, or we also call it the nanodrop. So this can be used to determine the concentration and purity of the DNA in a sample. So if you recall before, di ba nag-mention ko about the value kung sa ang purity of the DNA na nanodrop. So if it measures 1.8 ang atong nanodrop, then it means that atong purity of the sample is very pure or excellent in purity. But if it is greater than 1.8, it means nga there is a contamination, and the contamination is basically from the RNA. So maybe our DNA is mixed with some RNAs, and that will also that is also a form of contamination. So during sequencing later on, maglisod sa og pangita sa tinuod gyud nga sequence because there's contamination. And then here for the gel electrophoresis, it can be used to show the presence of the DNA in the sample and give an indication of its quality. So here's a picture na to, makita natin, no? That is an example of a um, DNA samples because of the presence of the smudges. So and unclear ng mga bands. So we call bands kind of mga line, solid lines that you can see on the mga solid bands that you can see here on the picture. So that those are the bands that dictates kung saan size sa atong DNA. Natin makita ang mga smudges, then that means nga dili clear or daay mga contamination ang atong DNA. So again, kaning presence of the DNA, we can confirm it by the use of electrophoresis. However, kaning electrophoresis, though it is widely used in molecular studies, this is very um, hazardous because it involves the use of lithium bromide, so it has a potential risk, no, sa atong health, and also it also uses UV light if we want to check it, if we want to check the fermentation of our DNA. So kinahanglan nato na siya i-subject to UV light because it reacts with the DNA. So makita na to ang mga bands with the presence of the UV light. I think last time we've already discussed about this DNA extraction from the breaking of the cell wall by the use of lysis to the dissolution of our DNA. So we can extract our DNA easier here today. Kaya na natin mga available ng mga commercial kits. So you can buy a commercial kit sa mga laboratories, and then they will already give you lysis buffer, wash buffer, elution buffer, and other mga buffers and mga chemicals that you will need, or mga reagents that you will need for the DNA extraction. So nanay ready to use. So even in the table that we have, then we can already conduct DNA extraction. So it's quite simple. It will already give you a good amount of DNA and a good quality. So pwede na tayo gamit. Though we will not be expecting no a very high quality like 1.8 yun, but the quality that it will give is already already on the range for good DNA quality. So, dili na ito problema. And it will be easier now to use or to conduct DNA extraction. Here also, we have another instruments nga ginagamit natin, no, in DNA extraction. And this is just a review because we already mentioned this last time. So, we can use mortar and pestle to break down the tissues, especially if we are dealing with kanang mga leaves or mga stems. So we want, of course, to grind it first, that we can use mortar and pestle. And this is also used for grinding, but it's a mechanical grinder where we use the beads inside the Appendorf tube. Then we also have here micropipettes. So not a single na micropipettes and multiple micropipettes. So this micropipettes is very much important, especially if we want to measure very minute amount of liquid. So if you want to measure in microliters, of course we cannot measure it in a test tube. Then we will use it, or we will use this, we call the micropipettes. So micropipettes have lahi-lahi ng mga measurements. So natay mga micropipettes nga kaya nga mo measure of uh, 0.5 microliters to 10 microliters, or napot tayo to 20 microliters, 30, 40, 100 microliters, and even 1,000 microliters. So, lahi-lahi nga mga measurement ang pwedeng nga ma-measure sa ato or magamit sa atong micropipettes. And this is the Eppendorf tubes. So, this Eppendorf tube is particularly important sa DNA mga storage of our DNA because this is where we store our DNA. So if you want to preserve it, then we can use this Eppendorf tube to store our DNA samples. And also, pwede sa uban yung mga liquid ng mga reagents kung gusto nato nga i-store, then we can put it in the Eppendorf tubes. So, laylay po ng measurements ang atong Eppendorf tube. So, na mga 1 ml, 1.5 ml, 2 ml. Another instrumentation that is used in DNA extraction, this one is the spectrophotometer. So, like siya nang sa usa, but this is the same older version lang siya sa atong spectrophotometer. So, namito na siya para makita ang quality sa atong DNA. And then we also have this one, the gel box. And this is used for gel electrophoresis. So, mara na siya duwa, ano. But this is very this is a very efficient tool or efficient instrument that is used in gel electrophoresis. So, there is atong gel box. Butang ka ng buffer. Later, I will discuss na lang later kung saan paggamit ang atong gel electrophoresis. And last one, this one is the centrifuge. So, during DNA extraction, dagan, di ba, I mentioned last time that there's a lot of centrifugation that will happen. So, diri na ito ibutang atong Eppendorf tube. And then, once it is closed, i-centrifuge na siya. Or there's an action wherein there's high revolutions per minute na may tabo diri Itoy-tuyok na siya until the solid will be separated from the liquid. So, basically, ang purpose why we do centrifugation para masettle ang solid and ang liquid is mas mapataas or mas ma-separate sa solid. So now, we're already done with the instrumentation. Now, I will introduce you with the polymerase chain reaction. This polymerase chain reaction is a technique that is used for us to create a lot of copies of a DNA segment. So, ang atong mga na-extract na before is not enough para for further nga mga processes or for further nga mga analysis. So, kato itong mga extract na from nga DNA, kinahanglan nato na siya nga i-amplify. Or when we say amplify, kinahanglan nato mag-create og dagan nga copy ana. And that is by polymerase chain reaction. And this polymerase chain reaction is developed in 1983 by Carey Mullis. So, ang gamit ano niya, imagine, you have only a single copy of the DNA or maybe hundreds of copies of your DNA gikan sa imong pag-extract. Then, from that 100, you can actually generate millions of copies of a small segment of your DNA or your specific segment na gusto nimo nga padaghanon because of that is your gene of interest. So, imong gene of interest, pwede na nimo padaghanon by the polymerase chain reaction. So here in polymerase chain reaction, the goal is to make enough of the target DNA region that it can be analyzed or used in some other ways. Say if you want to conduct gene sequencing, but you also want to conduct gel electrophoresis, but you also want to conduct cloning. So at least you already have the large number or large copy of your DNA polymer or your DNA sequence na imong imong interest or your DNA of interest. So they also call it as the molecular photocopying machine. So because it actually copies the template DNA nga imong nakuha from your DNA extraction. So again, from DNA extraction, imo siyang i-polymerase chain reaction para managhan. Kaya again, ang ato makuha from their DNA extraction is just small copies, small number of copies. So gusto natin na siya nga padaghanon para dili na ta magbalik-balik og DNA extraction. We want to do further analysis. So managamit sa atong polymerase chain reaction. And this also mimics the process of DNA replication. So, ma-recall natin sa itong Bio-32P sa uno or sa itong principles of genetics wherein ang DNA replication, it will provide a duplicate or a new copy for DNA. And this is also the same with polymerase chain reaction. So, wala niya gimimic ang DNA replication. But here in polymerase chain reaction, naalay certain nga segment or small segment sa DNA nga iyang copy. Not everything. Dili tanan sequences of the DNA nga i-copy. Only a certain amount. Kung kato lang ang imong interest, mao lang to ang i-copy. So, since this is a process, of course, nasa yung mga requirements sa atong PCR, and it is the same principle sa atong DNA replication na naitabo naturally. So, of course, if you want to replicate or if you want to copy something, then we need to have a template. Kinahanglan natin original copy before we can actually make a lot of copies in it. So, that's the DNA template. So, mao na siya, ang DNA template mao na ang imong gi-extract or a product sa imong DNA extraction. Next one is the DNA polymerase. So, sa function sa atong DNA polymerase, di ba? Atong DNA polymerase during the DNA replication, it is to elongate our DNA strands. So, siya ma-provide o complementary DNAs or complementary bases dito sa atong template to create new sequences. So, that is our DNA polymerase. Then, third requirement is the oligonucleotide primers. So, since small segments man lang ang atong gusto or atong interest, we need to create primers because these primers will dictate the polymerase where to start. Atong DNA polymerase, dili na siya maka-start o elongate or provide o complementary bases without our primers. So, ang atong one of the requirements is, of course, we need to have oligonucleotide primers. But here in PCR, we need to create our own primer. So later also, I will discuss how we are going to create our primers. Then we also have another one. The first one is the deoxyribonucleotide triphosphate. So of course, also nabitbiton sa atong DNA polymerase because this is in a machine. Kinahanglan kita po ang mag-provide sa atong mga nitrogenous bases. So, this is also another requirement for PCR. So, kaning atong deoxyribonucleotide triphosphate, bit-bito ni siya sa atong DNA polymerases. And lastly, the buffer system. Of course, kay mo-undergo man siya process, it needs to be, and the atong DNA, kinahanglan man nga dili madaot, so it needs to have an optimum condition. So we will provide a buffer system to provide optimum conditions for DNA denaturation and renaturation. So daghan man kayo nga mahitabo during the PCR. So we need to provide a good environment para sa atong DNA for it to maintain its stability and for it to denature and renature naturally. So this is very important for the fidelity, polymerase activity, and the stability of our DNA. So I mentioned, di ba, we need to have DNA polymerase as one of the requirements for PCR. And for DNA polymerase na to, specifically in the PCR or the polymerase chain reaction, we need to have Taq polymerase. So, kaning Taq polymerase is a polymerase that is coming from Thermus aquaticus. This is a bacteria that lives in hot springs and hydrothermal vents. And they are very heat stable and they are most active around 70 degrees Celsius. And at this high temperature, the human DNA polymerase or the E. coli will not be functional anymore. So later you will understand why we are going to use Taq polymerase instead of the DNA polymerase from human or the E. coli. So this Taq polymerase or the PCR requires DNA polymerase enzyme that makes new strand of DNA and it uses existing strands as templates. So mas ma-appreciate na natin siya later on if we look at the temperatures na kinahanglan nga ma-reach during PCR. So, this heat stability makes Taq polymerase ideal for PCR. Again, ang iyang pinaka-active na state is at 70 degrees Celsius. So imagine how high that temperature is. And kaya na niya nga mo live at this high temperature because naturally ang iyang natural environment is in hot spring and hydrothermal vents. So mao nang active siya at high temperature because diha man siya naturally exposed. So, man the importance ani ano man nga Taq polymerase man instead of other. I've mentioned earlier nga atong primers is that we need to create our own primers because in PCR we do not need to duplicate everything. Sa PCR dato nalatay gamay lang nga segment nga i-duplicate kadto lang ang atong interest. If we are not interested with that specific sequence, then of course we don't want to amplify it. Ang ato lang i-amplify ang specific sequence kung sa tong interest. Let's say for example we're interested with that specific sequence or a specific gene. Let's say kanang 1,000 ka base pairs lang ang atong interest. Then mo lang na pod ang atong ang atong gusto nga i-amplify sa PCR. So, because more na itong gusto, then we will create a primers. So, karang primers, they are, in the PCR, we need two primers that we need to use during the PCR reaction. So, we have first primer. So, unsa ito gamit ng primers? Siya ang mag-initiate or siya ang mag-initiate sa elongation process. And, siya ang mag-dictate kung asa dapit kinahanglan mag-start. And since duha ang primers na kinahanglan natong buhaton, makabalo pod kung asa ra pod kutob. No? So since kabalo man ang atong huan, naman tay giset nga mga primers 1 and 2. So it means nga diri ra mag-start. Dira ra mag-start and diri pod mag-end ang atong DNA replication nga may tabo sa PCR. So, kanila nga sequence or kanila nga segment diri ang atong i-amplify. So, mao na gamit sa atong primers. So, ang uban nga mga sequence nga naa pa dira, dili na niya i-amplify, dili na niya i-duplicate, dili na niya padaghanon. So, kanila nga atong region of interest ang atong kinahanglan i-copy. So, mao nang natay iset nga primer diri at the start and primer at the end. Para diha lang ko ito. So, mag-serve na siya as boundary sa atong region na kinahanglan nato may copy. So, the key ingredients of a PCR reaction, di ba? We have the Taq polymerase, the primers, the template DNA, and nucleotides. So, these four nga mga DNA building blocks will be assembled in a tube. So, natay gitawag nga master mix along with the cofactors that is needed by the enzymes. And they are put through repeated cycles of heating and then cooling that allows the DNA to be synthesized. So, now, this PCR has three basic important steps. First one is the denaturation. Second is the annealing. Then last is the extension. So in the denaturation process, kita natin nga, this is set at very high temperature at 96 degrees Celsius. So imagine nila nakataas. Ang boiling point sa atong water is 100 degrees Celsius. So init na ganyan na siya kaayo. So here, denaturation is near the boiling point of water. That's 96 degrees Celsius. Medyo makapaso na siya. And then we have the annealing at 55 to 65 degrees Celsius. And then the extension at 72 degrees Celsius. So, kung sa man day gamit ani nga mga steps. So, first step is the denaturation at 96 degrees Celsius. So, the sample containing the template DNA are heated to 94 to 96 degrees Celsius. And this is important because we need to separate or denature the double-stranded DNA strands. So, di ba initially, naatay double-stranded nga DNA kani siya. So, we need to heat it at high temperature at 96 degrees Celsius para mag-separate ang atong double-stranded DNA and it will become single-stranded nga mga DNA. So here, so how does it happen? So by that, the application of the high heat, magmelt ang atong hydrogen bonds that connects our double strand. So once nga magmelt ang atong hydrogen bonds, di ba ang hydrogen bonds man ang nag-connect sa atong nitrogenous bases that keeps the double stranded intact. So the goal of this first step, the denaturation, is to melt the hydrogen bond so that mag-separate ang atong DNA, double strands. So may mo siyang single-stranded nga DNA. Kita ni nato? Okay. So that's why denaturation occurs. So this is to provide single-stranded template for the next step. So again, denaturation is to separate our double-stranded bonds by melting our hydrogen bonds. Maalala na siya. And if we recall at our DNA replication, mura siya o gimimik niya ang process of the helicase na enzyme, di ba? Ang helicase enzyme nag-unwind sa atong DNA. So here sa denaturation, mura po niya gi-unwind ang atong DNA, but iya lang gi-melt ang hydrogen bonds. Second step is the annealing process. So here, the reaction temperature is lowered to 54 to 60 degrees Celsius for around 20 to 40 seconds. So, from here, kani siya, from kani diri sa denaturation, high heat, 96 degrees Celsius, nabuwag by the melting of the hydrogen bond. And now for the annealing, kinahanglan niya i-lower. So, syempre, kung padayon na gyapon nga 96 degrees Celsius, there's a chance na ma-degrade na hinuon na yung tanan nato nga DNA. So, kinahanglan nga ang reaction temperature i-lower to 54 to 56 degrees Celsius because this is also the time nga ang mga primers mo bind sa atong complementary sequence sa atong DNA template. So, at 54 to 60 degrees Celsius, ang primers mo attach. And by lowering the temperature, it will also stabilize our primers sa ilang pag-attach diri sa DNA template nato. So, i-attach ang primers. Diba? Ang gamit sa primers to initiate the elongation so that ang atong DNA polymerase will know kung asa siya dapat mag-start. So, primers bind to their complementary sequence on a template DNA. Primers are single-stranded sequence of DNA or RNA up to 20 to 30 base pairs. So, maghimong kitag primers, there's about 20 to 30 base pairs, but preferably, mga 20 lang itong gamiton ng mga complementary mga bases kapag himong natong primers. And this is the starting point for synthesis of DNA. Two primers are needed, the forward primer and the reverse primer. Again, and that is, again, to know kung asa mag-start, asa ra kutob ang atong amplify. Because, as I've mentioned earlier, when we conduct PCR, we don't need to amplify everything, every strand of the DNA. We only need to amplify our interest. Okay? So, kung unsa lang atong sequence of interest mo lang atong amplify, we don't need the other parts. Okay, for the directionality of our primers, of course, natay template DNA. And then, nabuwag yung atong template DNA because of denaturation. So, from kaning usa ka template, 5', 3', ang iyang complement atong mo run from 5' on this side to 3' here. 5' to 3', dire 3', 5'. So, it is always the same, no, sa even sa atong natural nga DNA replication. They are anti-parallel. 5', 3' from one strand, the other strand is 3', 5'. Same with this other strand. If 3', 5' ang template, atong primer runs from 5' to 3'. Last step of our PCR is the extension, it's the third step, and that the temperature is raised to 72 to 80 degrees Celsius. So, na-remember ninyo si Taq polymerase, di ba? They are very active and stable at high temperature of about 70 degrees Celsius. And that is also why during the extension, the temperature is raised at this high temperature because during the extension, ang pinakadakong trabaho dire is the Taq polymerase. It adds the complementary bases to the 3' end of the primer by the Taq polymerase enzyme, and it attaches to the primers and adds the DNA bases to the single strand. So during the elongation, ang atong DNA polymerase ang mag-add sa complementary bases sa atong DNA. So that is how these are amplified. So the DNA polymerase adds about 1,000 base pairs per minute. So in every minute, nga nakapaspas. So depende sa kung sa katas ang imong sequence of interest, then nga na sad kapaspas or kadaghan ilang ma-generate. So for example, ang imong sequence of interest is maybe 1,000 say 500. So in 1 minute makaproduce na ka og 2 copies of your DNA. Okay, from the DNA template, we separate ang strands through denaturation by high heat at 96 degrees Celsius. And then after that, the primers will bind to the DNA template. So, in an anti-parallel direction. So, ang katong 5', 3', atong primers will attach in 3', 5' direction. Sa 3', 5' nga template, ang atong primers will attach in 5' to 3' direction. So, after nga ma-attach ang primers at 56 degrees Celsius, erase na po ang temperature so that the Taq polymerase can act out and extend the primer. So, this primer, ang atong Taq polymerase will elongate by adding more complementary bases there. So, kutob na siya diri because diri man ang atong sequence of interest. Kung asa nag-start ang primer 1, mag-end sa siya dito sa primer 2. So, mo lang na ang atong sequence of interest. So, from template DNA, denaturation, addition of primers at 56 degrees Celsius, and then erase ang temperature by Taq polymerase because of the presence of Taq polymerase, which is very active at 72 degrees Celsius. So, erase po na itong temperature to 72 degrees Celsius para active and maayo ang performance sa atong polymerase and maayo ang addition of the DNA of the mga nitrogenous bases. So, this cycle will repeat at 25 to 40 times. So, initially, cycle 1, natay usa ka copy. Say, for example, initially, atong template, usa lang ka copy. After first cycle, create na tagduha ka copies. After second nga cycle, calculate na tao upat ka copies. Kaya mabuwag naman po din siya. Magbalik-balik naman tong denaturation, denaturation, annealing, and extension. So, after nga ma-create mo denature na po, magbuwag na po ng strand, ma-create na po another ng mga bases. And then, third cycle, then kani sila, mahimo na po ni sila nga, ma-separate na po ni. So, daga na kayo at the end of 40 cycles. So, maka-create or maka-generate tao millions of copies by the polymerase chain reaction. Okay, in your 1/4 sheet of paper, provide to me a summary of the polymerase chain reaction or the main events that happens during these three processes. During the denaturation, what is the temperature? What happens in the denaturation process? Also in the annealing, what happens in the annealing process? What is the temperature that is set in the annealing process? And lastly, in the elongation process. What happens in the elongation process? What is the temperature? And what is the primary enzyme that is involved in this, or what is the primary Yes, what is the primary enzyme that is involved in this one? And also in your 1/4 sheet of paper, please write the importance of polymerase chain reaction. Okay, next we will proceed with the gel electrophoresis. Suppose you have just done a PCR reaction and you already created many copies of a target DNA region. Now you want to check and see whether your PCR worked and the technique that you can use to visualize and confirm the size of the DNA. So this electrophoresis involves running a current through a gel that contains molecules of interest. So this is a technique that is used to separate our DNA, our RNA, and protein based on their size and charges. Um, ang importante lang yun diri sa gel electrophoresis is that you should have a good amount of your DNA. So, if gamay lang yung DNA sample, don't mind about using gel electrophoresis. Or, I mean, don't think about using it because wala kayo makita. So, ang prerequisite sa gel electrophoresis is that you should first do PCR reaction before ka mag-gel electrophoresis because you need to have hundreds of thousands of copies of the DNA para ma-detect na siya sa atong gel electrophoresis. So, our electrophoresis involves running a current through a gel that contains molecules of interest. And makita nato sa ang gel electrophoresis, or this is very important because we will be able to know kung unsa ang size sa atong DNA. Here on the picture, there is a tulip. This is an example of a good uh, and pure DNA. So, kita ninyo, clear kaya mga bands. So, these are the bands, ang mga lines. So, kaning mga bands, mo na siya mag-dictate sa fragments sa atong DNA. Here, for example, kani siya. So, kani sila, makayunta kaning samples 6, 7, and 8. They are of the same size, basically. So, this one, kaning na diri sa atong 1, well 1 and well 12, kaning na one, diba? We can see nga, medyo daghan sila og linya. So, these are what we call the smart ladders or the DNA ladder. Kaning DNA ladder, pre-made na siya. And this is also very important when we conduct gel electrophoresis. We need to make sure na natay DNA ladder. So, kaning DNA ladder is very important because this already has the kind of pre-made na nga fragments. Makabalot nga, okay, kani siya is maybe at 1,000 base pairs. So, here. So, di kayo maklaro ba? Siguro, kani, this is already at 1,000 base pairs. This is at 100 base pairs. So, kung kinsang motungod na niya, say, for example, kani siya nga ladder is 500 base pairs ni siya. So, kung tungod din siya, siguro makayang nga, this is also 500 base pairs. So, ang asang iyang tungod mo na ang iyang exact na size. So, again, ang electrophoresis nag-involve siya running a current through a gel containing the molecules of interest. So, from the electrophoresis, of course, we need a current from that. So, gel electrophoresis, gels for DNA separation are often made out of a polysaccharide, agarose. So, ang atong medium, sudlanan nato sa atong DNA are what we call the agarose gel. And this is from an agarose powder. Kinahanglan lang nato adan o buffer solution para mahimo siya nga semi-solid. It will be a gel-like, medyo gahi nga gel na structure. And this gel has packet-like indentations. We call the wells. So, nanay buho diri sa gels. And then, we will put our sample here. Diri sa wells. Of course, we need not to forget the ladder. Butangan gino natong ladder always sa pinaka-first. Kaya mo natong basihan sa size sa atong DNA. So, after nga ma-set up na nato ang atong mga samples diri, butang na nato siya sa current. So, mo flow ang current by the presence of the buffer. So, diri ang atong gel is submerged na siya sa buffer. So, this is the buffer kaya siya ang blue. So, mo ni ang buffer. This is a liquid solution wherein diri mo agi ang current. So, we have the positive electrode and the negative electrode. So, since ang atong gel electrophoresis kinahanglan man current, kita natin dire this is negative electrode, di ba? And this is positive electrode. So, ang movement sa atong current is going to the positive electrode. So, nga naman, kinahanglan nato nga ibotang diri ang atong wells dapat duol sa atong negative electrode. So, always na when we put the when we put the gel, dapat ang atong wells is facing on the negative electrode or mas duol towards the negative electrode as compared to the positive electrode. So, ano man eh. So, if you recall, ang atong DNA is rich in a phosphate group, di ba? Because it is its backbone. And this phosphate group provides a negative ion diya sa atong DNA. So, negative, kinahanglan nato nga padaganan ng atong DNA. So, dapat attracted siya sa positive electrode. So, manang atong DNA, if you want to know the size, kinahanglan siya nga mudagan padung sa positive, di ba? Opposite attracts. So, positive, moduol yun na always sa negative. So, magpalayo siya sa negative electrode, paduol siya sa positive electrode. And that is how we start to that is how we determine the size of our DNA. So, kung mas gaan ang atong DNAs, let's say for example 300 base pairs, so most likely i-expect nato nga ang flow or ang daga niya is faster kaysa sa atong DNA nga mga bugat, like say for example 1,000 base pairs nga DNA. So, mas hinay ang iyang dagan kaya mas bugat man siya as compared sa katong mga gaan which is 300 base pairs lang ng mga sequences, then pas-pas ra na ang iyang dagan. So, what are the requirements kinahanglan nato for gel electrophoresis? Of course, we need our sample, DNA sample. But this sample kato ng amplified nga sample, kato ng gikan sa PCR because we need hundreds of thousands of copies sa atong gel electrophoresis para ma-run sa atong gel electrophoresis. We also need a current. So, kinahanglan nato kurente. Kinahanglan nato electrical pulses runs from negative to positive. So, ano kaya itong negative to positive? Because we want to know the fragments. We want to know the size of the fragment. Ang atong DNA is negative ang ions niya. So, it needs to be attracted to the positive. So, it needs to run to the positive direction or the opposite direction. And then we also have here the agarose gel. It provides space or work as a vehicle to run DNA under the current. And then we also have buffers. So it's also a medium that facilitates easy migration and also the hapon mo flow ng atong current sa buffer na to. And the dyes. So we want to visually see, gusto na ito makitaan yun, no? Kung unsay dagan sa atong DNA. So we want to put dyes to track and monitor the migration of our DNA. Also here, sa atong when we conduct gel electrophoresis, kinahanglan po din natong bantayan ang time. Say, for example, i-subject na to siya no, to gel electrophoresis, di ba? May negative and positive electrodes. As ang well dapit, dool sa negative electrodes. So, most commonly, ang atong gel electrophoresis mo dagan na siya about 30 to 40 minutes. If malapas na siya 30 to 40 minutes, there's a high chance nga ang imong DNA samples mulapos na siya sa imong gel and masayang maglutaw-lutaw na siya sa imong buffers. So you need to be careful mga matanaw ka, especially if kabalo ka nga gaan ra ang imong DNA or imong interstranded base pairs lang. So, at 20 minutes, tanaw na nimo base na lapas na ang imong DNA. No? So, pwede na siya mulapos na siya diri. So, how do these DNA fragments move through the gel? So again, we have the DNA ladder. You have the DNA ladder, and diri mo ipambutang yung mga DNA samples, no? And these samples are loaded into wells. Then the power is turned on, and the DNA fragment migrate through the gel. So this is the negative electrodes, no, diba? As you can see, mo niya itong negative electrode na diri po duol ang atong wells. Kung asa dapit ang negative, dito dapat mag-face ang atong well. And opposite siya sa positive direction. Sa imong current or sa imong gel, madya ang atong mga sequences. Let's say first nga DNA sample nato is kani siya nga kuan kani siya. Say ayan ta kani siya is mas bugat as compared to here. Kay pas mo ni siya mo dagan. So mabala nato nga pas siya mo dagan is if duol na siya sa positive electrode. So moduol man na sila because of the mas mo attract man sila sa positive. So mo nang magpalayo sila sa negative charge. Mo ra gud og magnet. Nga negative is dili gid siya mo work. But negative positive mo pilit sila. So that is the same principle nga naa diri sa atong PCR. Sa PCR sa atong gel electrophoresis. So ang negative ions from the phosphate group sa atong DNA ma-attract na sila sa positive charge. Mauna nga matikaduol sila. So mas gaan sila, mas paspas ang ilang movement. Sabtan, mas bugat sila, sina ilang movement. So diri ayang tanga mo ano yung pinakadako ng largest sequence or largest number of DNA sequences. Kaya napo man siya diri at a, let's say, specified time. Let's say, for example, after 30 minutes of gel electrophoresis. Kaya mo na niya itong nakitaan. So, kita na ito nga, maybe this is largest. Kaya naman siya, duol pa man siya sa negative, hinay man ang iyang dagan. This one, this may be the smallest. Let's say, maybe this is 300 base pairs. Kaya, paspas siya mulagan. So once the fragments have been separated, so we can now examine the gel and see what size of bands are found on it. When a gel is stained with a DNA binding dye and when they are placed in the UV light, same as here, the DNA fragments will glow and it will allow us to see the DNA that is present at different locations along the length of the gel. So we can see here a well-defined line of the gel and we call this the band. Kani, this is the band. This is what I've mentioned earlier, the DNA ladder. So this is already pre-made and this is standard size molecular marker or fragments that is of known length that is used to determine the size of the PCR. Usually the PCR products are run to determine the fragment size. And different fragments have different running speeds. Larger fragments will run slower, as I've mentioned earlier, while the smaller fragments will run faster. Mas paspas sila maabot dito sa positive na electrode. So technically, without a reference or standard size marker, we really won't understand kung size ana nga fragment. So without any reference, the gel run will not also make sense. So without this DNA ladder, ang atong pagpadagan sa gel electrophoresis will not make sense because we will not know kung saan size yung fragment. And that is basically why we are doing we are doing gel electrophoresis for us to know the size of the fragment. So without this, we won't be able, or this will not make sense. Without ladder, kung wala tayo ladder, wala gamit ang atong pag-run sa gel electrophoresis. Diba? So the DNA ladder is simply a composition of a standard sized fragments which runs according to the fragment size. And this will actually help us to determine the size of the DNA fragments. So always nga when we conduct gel electrophoresis, at least one well is reserved for a DNA ladder. And that is a standard reference which contains the DNA fragment with known lengths. So mga commercial ng mga DNA ladders may come in different size ranges. So we would want to pick one that has a good coverage of the size range of our expected fragments. So say for example, atong fragment is 5,000 ang atong DNA fragment or atong interest. So dili ta mo gamit og DNA ladder nga kung lang 1,000, mo gamit gid ta og mo range siya og about 8,000 because atong goal is na may 5,000 nga atong expected nga fragment is 5,000. So ana to siya paggamit sa itong ladder. Kaya lahi man na may mga ladders na 1,000 lang. So if ang imong segment is or ang imong sequence of interest is only at 500, then pwede na mo gamit og 1,000 nga ladder. So once nga ma-separate na nato no ang fragments, we can now examine the gel and see kung saan size ang naana nila. So a well-defined line of DNA on a gel is called a band, which I mentioned also earlier, and each band contains a large number of DNA fragments of the same size. So dari no, nato. So first, this is maybe about kaning first siguro nga sample, siguro this is about 4,000 or 5,000 base pairs. Kaya bugat siya kayo. So at 30 minutes, say for example, at 30 minutes mo niya nga itong result. Kini, siguro this is about 150 base pairs. Ni, this is 500 base pairs. Mawala ang akin nga, kini day akong din example. Sakto di ay, akong gi-expect kay 500. O niya, natunong siya diri sa nga 500 gid siya. So, siguro, sakto gid ni. Makaingon ta nga, medyo sakto niya nga to kuan. Sakto niya nga to, gi-amplify nga DNA. Because there are times, no, na kanang ang imong gi-amplify is dili day mao. Maybe na sayo pang imong primers or sayo pang imong mga reagents. Dunay times nga lahi ang atong kung ang atong expect is 500 tapos may buong na lang ta. Anong 1,500 man yung mga na-view ani nila. So this can be coming from a different DNA rather than our gene of interest or our DNA of interest. So here, also in your 1/4 sheet of paper, based on the results of the gel electrophoresis, answer this one. What is the estimated length of the fragment? Fragment B, what is the estimated length? Fragment D, what is the estimated length? And fragment C, what is the estimated length? Another question, which DNA fragment is the shortest? Which DNA fragment is the longest? Kindly answer it in the 1/4 sheet of paper. Okay, last, 7 samples are run on agarose gel electrophoresis for 6 different markers. Marker A is only present in sample 2, 3, and 7. Here is the question. A scientist purified samples of DNA from several puppies. She expected to determine if there are any twin puppies in the group. The image depicts an agarose gel in which a set of DNA size reference standards were electrophoresed alongside the scientist's experimental DNA fragments. Now, based on the results of this analysis, are any of these puppies likely to be identical? So, which of the following samples are the identical puppies? Is it from these samples 2 to 7? Asa kaani ang identical puppies? Again, write it on your 1/4 sheet of paper. And there's still other mga questions pwede ninyo ma-practice. Now, the result of agarose electrophoresis are affected by some factors that are enlisted below. So there are actually some factors that affects the result of our agarose. So first is the concentration of the gel. If ang atong concentration sa atong gel is sobraan sa kakumo, then it will also affect the result of our DNA electrophoresis. So dili makatarong flow ang atong ang atong DNA fragments. Or maybe if gahi na sad siya kayo, so dili dyan maka-penetrate or maka-migrate ng atong DNA. Another factor is the reuse of chemicals and solutions. So there's very high tendency if we reuse chemicals and solutions nga magka-cause na ng contamination. Imbis pure ang imong DNA, nigamit ka og mga chemicals nga nagamit na sa una, then there's a high chance of it nga maka-cause of contamination. Another is unpure nga DNA sample. So kung dili na pure imong DNA sample daan, do not expect nga nindot ang imong gel electrophoresis. So that is why I told you earlier or before pala sa kuan. I've told you in the previous lecture na DNA extraction is very crucial. Kasi if dili pure ang imong DNA sample, dagan og kuan, dagan og contamination. So sa possibility sa imong gel electrophoresis ma-detect po na siya. May bong nga kadaghan na kay kag bagis sa imong DNA or sa imong gel electrophoresis daghan na kay kag bagis. So kabalo ko kasi tinuod ano. And then the concentration of agarose dyan. So I mentioned din natin kanina. So there's still also a lot of other more factors about gel electrophoresis na maka-affect sa atong gel electrophoresis. Okay, that's all for today. Mo lang na siya nga atong topic for today, and don't forget about tech quiz. I will get it next meeting. So on a 1/4 sheet of paper, katong question or summary on the PCR and also 2 slides nga questions dito sa atong gel electrophoresis. So if you have questions, you can PM me or you can send message to our group chat. That's all. Thank you so much.